All right, hi everybody. Good. All right, so as promised, we're going to do a couple of examples dealing with RNA and the genetic code. So first off, let's just talk a little bit about the different types of RNA that you come across. And there are essentially three main types of RNA. Let's go to color. All right, you have your ribosomal RNA, which is commonly known as rRNA. You have your messenger RNA, which is what is commonly known as mRNA. And then you have your transfer RNA, which is commonly known as tRNA. And essentially, all three of these types of RNA are essentially involved in protein synthesis. Now, just like in the notes, in a sense, we're not going to go through every single aspect of protein synthesis. We'll talk a little bit about transcription and essentially translation, the two sort of main steps associated with it, and we'll provide some details, but we're not going to go through the entire process. When I think I spelled ribosomal RNA wrong, so let's just fix that. So I had too many S's. Ribosomal RNA. So from my perspective, in terms of the three different types of RNA, the two main players in protein synthesis are really the messenger RNA and the transfer RNA. And as we'll talk about in a few minutes, one of the things to understand about protein synthesis is the idea that the messenger RNA calls for the amino acids to be brought to the ribosomes for protein synthesis, but the transfer RNA actually brings it. And we'll talk a little bit about the relationship between messenger RNA and transfer RNA as well. So essentially, to talk a little bit more about the three different types of RNA, let's take a look at an example question. All right. So in this question here, what we want to do is we want to match the following statements with ribosomal RNA, messenger RNA, or transfer RNA. All right. So letter A carries genetic information from the nucleus to the ribosomes essentially for protein synthesis. So essentially, which one of these three types of RNA does this statement refer to? This statement refers to messenger RNA or mRNA. So essentially, what you have is you have a DNA segment that unwinds. And a portion of that unwound section of essentially one nucleotide strand is used to make a messenger, RNA. All right. And again, in going back to DNA for a second, each DNA molecule is a double helix that has two nucleotide strands winding about each other like a spiral staircase. As we talked about previously, the force of attraction that you have between the complementary bases on the strands is hydrogen bonding. That force of attraction holds the strands together. Well, essentially, an enzyme is going to break a hydrogen bonding in a portion of that DNA double helix. And as we'll sort of get into a little bit, there's going to be sort of an identification point of where sort of messenger RNA, where the messenger RNA should start to be made from in a certain sense, okay? So you have a DNA segment that unwinds by breaking the hydrogen bonds between complementary bases, not the entire DNA double helix, but a segment of it. And then a portion of that unwound section of one nucleotide strand is used to make a messenger RNA. Now, what's the relationship between DNA and messenger RNA? DNA and messenger RNA are complements of each other. And this is really very similar to the idea of the complementary base pairs that you have in the DNA double helix. Essentially, if you consider cytosine, guanine, thymine, and adenine, the ideas of complementary base pairs that we see in the DNA double helix hold true, except for one big difference. So if your DNA has cytosine, your messenger RNA is going to contain guanine. If your DNA contains guanine, your messenger RNA is going to contain cytosine. If your DNA contains thymine, your messenger RNA is going to contain adenine. But if your DNA contains adenine, your messenger RNA is going to contain uracil. This is the difference in terms of comparing complementary bases in the DNA double helix, because RNA doesn't have thymine. Instead, RNA has uracil. So this is the one difference. And we'll apply this to a question that we'll do in a little bit. All right. So that's the first statement, letter A. And some little bit of other information about it. All right. Letter B. Brings amino acids to the ribosomes for protein synthesis. So as we mentioned at the beginning of the video, your messenger RNA calls for the amino acids to be brought to the ribosomes for protein synthesis. But what actually brings the amino acids to the ribosomes for protein synthesis is your transfer RNA. So your transfer RNA would be the answer here. Now, what's the relationship between messenger RNA and transfer RNA? Well, messenger RNA and transfer RNA are essentially complements of each other. So just to sort of expand a little bit, on the table above, we can fill in for transfer RNA. If your messenger RNA contains guanine, your transfer RNA is going to contain cytosine. If your messenger RNA contains cytosine, your transfer RNA is going to contain guanine. If your messenger RNA contains adenine, your transfer RNA is going to contain uracil, right? No thymine. And if your messenger RNA contains uracil, your transfer RNA is going to contain adenine, because messenger RNA and transfer RNA are complements of one another. Letter C. combines with proteins to form ribosomes. Combines with proteins to form ribosomes. This would be your ribosomal RNA, or your rRNA. Combines with proteins to form ribosomes. This is your ribosomal RNA. Letter D. Acts as a template for protein synthesis. And essentially, the template where what essentially we look at to figure out what amino acids are going to be brought to the ribosomes is going to be your messenger RNA. And then finally, letter E makes up the highest percentage of RNA in the cell, and essentially the most abundant type of RNA that we have is the ribosomal RNA. All right, so hopefully just by looking at these questions and with a little bit of extra in-depth information, we now have a pretty good idea of the 3 different types of RNA that exists. All right. So as I mentioned a little bit earlier, the 2 key steps in protein synthesis are first transcription. And essentially in transcription, DNA is made, or I should say, let's say it this way. Messenger RNA is made from a section of DNA. Remember, one of the key differences between DNA and RNA is that RNA tend to be much smaller than DNA. And one of the ways I always say that you can rationalize this is based on the fact that messenger RNA is made from a section of DNA. So it's one way to rationalize it. The other key difference, besides the other ones that we've talked about in the previous video we did, the previous Zoom session, is that RNA molecules are single-stranded versus DNA molecules, which are double-stranded. That's the final key difference. So in transcription, messenger RNA is made from a section of DNA. And then essentially you have your translation, where the messenger RNA calls for amino acids and we use what is known as the genetic code to interpret the sequence of bases in sets of 3 called codons. And then your transfer RNA brings the amino acids to the ribosomes. So again, this is a key point. The messenger RNA calls for the amino acids, the transfer RNA brings it. But how does the messenger RNA call for the amino acids? Essentially, the bases that are found in the messenger RNA, because remember, they're made up, just like DNA, they're made up of nucleotides. So you're still going to have phosphate, sugar, and base. Just like we mentioned how if you think about DNA, the only difference between nucleotides would be in that DNA would be the nitrogenous base present. Same thing for RNA. The only variable that you have in going from one nucleotide to the next in an RNA molecule would be the nitrogenous base. In what is known as the genetic code, bases are not read individually. Bases are not read in pairs, they're read in sets of 3. And each set of 3 is what is known as a codon. Now, in the notes and in your textbook, you have the genetic code. You don't have to memorize it. For the final exam, I will give you a copy of the genetic code as well to solve problems. You don't have to memorize it. Couple points. First off, if you look at the genetic code, there are a total of 64 codons. There's only a total of 20 amino acids. So as the genetic code easily illustrates, since basically we have triple the number of codons than we have amino acids, almost all amino acids have multiple codons that code for them. Then we also have the start codon. Our start codon is the signal for protein synthesis to begin. And there's only one of them, and it is AUG. And what you notice is that it codes for methionine. So essentially, as the protein is being made, for every instance, the first amino acid present is going to be methionine. But by the time all is said and done, that initial methionine that's found is usually removed. But as essentially the protein is being made, the first amino acid that you're going to come across is going to be methionine. But again, before all is said and done, then basically that initial methionine at some point is going to be removed. And then on the other hand, you have your stop codons, which signal protein synthesis to end, because they do not code for any amino acid. So no amino acid is going to be brought by the transfer RNA. And that's the signal for protein synthesis to come to an end. Which are the stop codons? You have, and that looks too much like a V. So you have UAA, you have UAG, and you have UGA. And you don't have to memorize these. You know, it's clearly illustrated on the genetic code which ones are the stop codons and such like that. All right. So you have transcription, where you have the messenger RNA being made from a section of DNA. And then essentially you have translation, where the messenger RNA will call for the amino acid to be brought to the ribosomes, and the transfer RNA will bring the amino acids to the ribosomes to make the protein. Again, we're going to keep it very generalized. You know, we're not going to get into all the specifics in terms of essentially initiation, chain elongation, things of that matter. We're going to just keep it to a very broad level. So let's take a look at an example. Give the corresponding messenger RNA, transfer RNA, and amino acid sequences for the following segment of DNA. erase the whole thing. All right. So actually, let's do that. Let's do that example again. And let's do it the right way. So DNA is going to contain A, T, G. Then we've got C, C, A. Then we've got C, C, T. Then we've got A, A, A. And then we've got A, T, T. All right, that's better. Hopefully you caught that mistake, but if not, there it is. And I'll see if I can actually edit the video to take out that problem, but we'll see what we can do. All right, so we got messenger RNA, then we've got transfer RNA, and then we've got the amino acids. All right, so we'll have, remember, DNA, messenger RNA, complements of each other. So we've got U, A, C, G, G, U, G, G, A, U, 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 A, A. All right, now let's go from the messenger RNA to the transfer RNA. So uracil would give us adenine, adenine would give us uracil, cytosine would give us guanine. Guanine would give us cytosine, uracil would give us adenine. Remember, messenger RNA and transfer RNA are complements of each other. 2 guanines would give us 2 more cytosines. The adenine would give us uracil. The 3 uracils are going to give us 3 adenines. And then finally, the UAA will give us AUU. All right. The amino acids. And this is really, when I graded the homeworks a little bit, I've seen the mistakes. Because what some students are doing is they're using the transfer RNA. No. When we use the genetic code, remember, the genetic code is based on the messenger RNA sequence. So we're not going to use the transfer RNA sequence. We're going to use the messenger RNA sequence. All right. So I'm looking at the genetic code on another screen. I find UAC. And when I come across UAC, and the 3-letter abbreviations are perfectly fine. I have no problem with that. So UAC gives us tyrosine as our first amino acid present. GGU would give us an amino acid of, sorry, not GLU, would give us GLY. And let's get the pen back. Okay. But it's still there. Let's get the pen back. All right. So it'll be GLY. I don't want subtitles either, but let's see if we can figure out. There we go. Knew it had to be something I hit. GLY, which of course is glycine. GGA would also give us glycine, and I still didn't get the pen back. There we go. Would give us glycine. So we would have 2 glycines back to back here. UUU would give us phenylalanine, PHE. And then UAA would actually be a stop codon. So we would have a signal to come to an end. All right. So tyrosine, glycine, phenylalanine, and then stop. And again, the key in terms of determining the amino acid sequence was that I used the messenger RNA. Right? So don't use the transfer RNA, use the messenger RNA. And again, you'll have a copy of the genetic code, so you'll be able to use that to figure out the amino acids, and the 3-letter abbreviations are perfectly fine. All right. So I hope these couple of problems help, and I wish everybody